the milk looks rancid. It is very, very foul. And so you'll see like the milk separates and this kind of stuff. So it and it looks really a really off yellow. So hopefully anyone who engages their brain will be like, hang on a second, that looks absolutely dire.
All right, welcome everyone to another episode of the Bioinformatics Lab Podcast. I'm Kevin Libuit with Andrew Page. We're from Fusion, and today we're going to talk about an ongoing current outbreak of H5N1. This is obviously huge news across North America, devastating a lot of our poultry and now even our cattle agricultural practices in the U.S. And there's also been some concern over there on the European continent about, the potential spread from the North American flocks to Europeans. Andrew, what's your understanding of things right now? If you can provide some context to folks about, in general, the public health landscape of H5N1.
So maybe just start off, H5N1 is an avian influenza, and it's a highly pathogenic avian influenza. So obviously that means it spreads quite easily. And, that generally there's different types around. There's ones that affect birds, there's ones that affect, like, say, swine and, other animals. And then there's ones that affect humans. And the problem, I guess, with influenza in general is that it's really, really good at recombining. And it'll take little bits from here and there and everywhere, and then come up with yet another magic influenza to, run riot across the world. And that's always what people are afraid of, this pandemic X. I know we've had coronavirus, SARS-CoV-2, but actually the one that people are most afraid of is actually influenza, because it can have devastating effects like the, Spanish flu back over more than 100 years ago, and things like that, regular waves of challenges. So in the US, there's a big outbreak in birds of this H5N1, and of course, birds are a difficult problem because you've got wild birds, you have birds used for food production, poultry, and then of course that can spill over then occasionally with some small mutations. These particular strains, H5N1, can spill over into other populations, like in this case cattle.
Yeah, that's right. And I think what you brought up about SARS-CoV-2 is so interesting, because I feel like, 10 years ago or something like this, I would find myself going into conferences, and there's always those sessions about like the pandemic potential of infectious diseases. And there's always two broad categories. It was either the antimicrobial resistance conversation and the dissemination of resistance genes across the world, and then influenza. And because of exactly what you talked about, reassortment is intense. For those of you who are unfamiliar, it's a multi-segmented genome, and those can hop around, especially in these co-infections, to create exactly what you said, these magic influenzas where, I think in like early 2000s, there was the swine flu pandemic across Southeast Asia that, was devastating throughout the population because it was a recombination of 3 different types of influenzas. And there was a human strain, a avian strain, and a swine strain, all of which recombined to create this like intense bug that wreaked havoc on the population. So that's always the concern whenever you're seeing a very infectious influenza strain circulating throughout the global population. And in this case, I didn't know this, by the way, highly pathogenic avian influenza. That highly pathogenic designation is with respect to the severity of disease in poultry. I just always, am very human-centric. I thought it meant like HPAI meant it was going to be high disease severity in humans. Not necessarily the case. So when looking at avian influenza, right now it's, the dominant strain is H5N1 across the bird population. And in the US, it became a huge deal because right now it's destroying like a lot of our poultry farms. And I think U.S. audience are going to feel this immediately. This is something that you can even see at your grocery stores. Literally eggs are no longer on shelves across the U.S. Yeah. It's a big deal. And again, as pandemics start to impact your lives, like I'm walking through the grocery stores. I was like, Costco the other day, and you go into like this special section for eggs, and it's nothing, no poultry eggs. There's like, quail eggs or something like that. People try to find alternative sources, but no chicken eggs. Interestingly, no shortage of chicken meat. Different bird populations. There's like egg-laying populations, and then they call it broiler chicken population. So this is particularly so as in terms of pandemic potential. This is a disease that's already impacting the poultry farms across the U.S. And it became an even higher level of concern because you're seeing the mammalian spillover in the U.S. So from the bird population to the cattle population, of course, that gets everyone's alarms up in terms of, okay, now it's in the mammalian population. Is it going to have that adaptability to human population? And there's been not many, but there have been observed cases of H5N1. I think still less than 100 relatively mild cases in the U.S., save for a couple of severe cases. I think there's now 2 severe cases in the U.S., one of which led to a fatality associated with H5N1. So, yeah, there's a huge impact in the U.S., and I think there was also a risk assessment from the European scientists, addressing this.
Yeah, I guess in terms of spillover, there were multiple identified spillover events from birds into cattle. So you have is it B313 and
Nice,
D11.
yes.
And because these lineages are distinct genetically, you can actually that their spillovers is not just transmission between cattle. So it happens all the time, unfortunately. And with the paragenomics, we can actually identify this, and we can classify it, and we can understand it much better than we have before. I guess one of the risks and limitations is that obviously with the changing government, the CDC has reduced its outputs in terms of regular updates to surveillance data. So that is kind of a risk. We don't necessarily know what's fully going on, but at least states are not bound by that, and they are alerting the public when there is different issues. So in the UK, I sit on an advisory committee with the Food Standards Agency, the Advisory Committee for the Microbiological Safety of Food. And we received a risk assessment on what the potential impact on food is, from products coming in from the United States for H5N1. And the answer is, the risk is very low, to be honest, to the UK from influenza spreading through that route. And mostly it's because there's not very much coming into the UK. So, very little poultry and dairy products, meat, and, it's just not a thing. And, some risk could be like dried milk powder, where obviously when you freeze dry a virus, sometimes it can kill it, it's and sometimes it's super good at preserving it. And that's where you get your kind of live attenuated viruses, in powder form, and they can be reconstituted because it's really good at storing some viruses. And so there's that kind of uncertainty with the process. But hopefully, in theory, all of the milk going into, say, powdered milk or, say, baby formula is pasteurized before export, before being freeze-dried. So that should mitigate it more or less, we hope.
That's an interesting point to bring up, pasteurization. Because what you're seeing now in the U.S. is cattle population has been impacted. And in the highest risk is obviously agricultural workers who are dealing directly with these infected animals. Because actually the means of spread is by the dairy products, which I thought also was pretty fascinating. It's not, I just anticipate like the respiratory side of things, but it's the dairy products themselves have, live flu virus.
Oh, they are.
That's
The milk is absolutely jam packed. It has extremely high levels of virus. When a cow's sick, extremely high levels of virus. But the good thing is that the milk looks rancid. It is very, very foul. And so you'll see like the milk separates and this kind of stuff. So it looks really a really off yellow. So hopefully anyone who engages their brain will be like, hang on a second, that looks absolutely dire. But then, you usually with milk, it'll be mixed into big vats and whatever, so it might be diluted out.
That's exactly right. And so on the dairy side, a lot of the cases have actually been mild, like conjunctivitis. So getting it into, the conjunctiva, I guess, from the dairy workers. But then also, there's been like a big feline outbreaks from unpasteurized milk products as well. And that's a big sort of conversation in the US about pasteurization and things like that. So hopefully
Yeah.
another more fodder to the argument of pasteurization being a strong public health practice across the US. But yeah, so that's I think a pretty good outline of what's going on in terms of the outbreak. And I have had to constantly tease about the nomenclature around this, because when we talk about H5N1, that's influenza A typed on the H and N proteins, but then there's the conversation of the specific clade. So looking at the genetic data that is hyper relevant, I forget, it's like 2344B or whatever it is the clade, and then further classification of the genotype that we're looking at in B313 that's impacting the cattle population, and then the flock population on the D1.1. So a lot of the surveillance efforts have to take into consideration those different subtypes, because now what we're seeing in public health labs, a lot of federal agencies in the U.S., including the CDC and USDA, are starting to generate sequence data for these samples. And whenever you're doing, for example, an assembly, a lot of it is reference guided. So you have to have an understanding of what reference to select, and then how to do the genomic epidemiology too. Again, a multi-segmented genome like this, there's a lot of consideration. So it's actually kind of fascinating to watch the discussions in this space, because traditionally, when you're looking at influenza phylogenetics, it's only HA segments, and you're looking at the evolution of the HA segment. But in this case, you're starting to see a lot more full segment, whole genome trees. So they just concatenate all the segments based by, again, such fascinating things, because you have to think about how you're going to concatenate these genomes. So like in general, the practice has just been size concatenation. So like PB2 is like the largest segment. So you start with that, and then whatever, it cascades down from there. But you're seeing a lot more of those trees being utilized and made available to the space. For example, I think it's the Moncla lab in the US. They've done a huge effort in maintaining a concatenated H5N1 tree. Angie and Russ Corbett's team over at UCSC and their Usher builds have made D1 and B313 trees available. So there's a lot of interesting bioinformatics happening here too.
And you might notice that the nomenclature of the different lineages is the same as SARS-CoV-2, which is really handy because, it's quite a simple way of naming things and numbering things and capturing all that diversity that's out there.
The viral world. Yeah. And there's been pretty good, participation across at least the technical community. And I know in the U.S., we're trying to work very closely with the state public health labs and even federal partners and academic partners to figure out what the best practices are. We learned this from SARS-CoV-2 of just having forms of communication really help to understand, hey, what are the blind spots? What are the best practices? What are even the tools people are using to make analysis possible? So, and because it's a virus, a lot of the same major players that were helpful in understanding SARS-CoV-2 have also already started contributed heavily to understanding H5N1. So namely the next strain universe of practitioners from like making NextClade resources available for proper typing and mutation profiling and all the rest have stepped up in a really major way for H5N1.
And actually, this the monitoring of all of these genomes and sequencing of them does help quite a lot when it comes to vaccine development. Like you can see the annual vaccines that come out for flu. They change every year depending on what's circulating in the other hemisphere. And you can make a flu vaccine very rapidly. I think there's a CDC study a few years ago where they went to one of these pig fancier shows. Do you remember that? And,
with like a MinION or something too?
Yeah, they went around swabbing all the pigs, and they went sequenced them, on site with a MinION, and then created a vaccine within a week. Like, that's serious science, And I think the guy who led that is now working for the AMD now and CDC. Mm-hmm. So, like, seriously good science. And it just shows how quickly you can get a vaccine out if you know what's going on, and the power of sequencing there, to actually tell you what is there and how, guide you in those decisions.
Yeah, absolutely. The sequencing applications are vast in this kind of emerging outbreak situation. As you know, obviously we're biased, but as it could be applied to any infectious disease outbreak. But not only on the vaccine side, you're seeing a lot of the conversation of monitoring for key mutations. Like that's what we're working with different laboratories. Like so, there's already been a lot of laboratory studies of okay, what mutations would be indication that we're watching mammalian adaptivity or human transmissibility kind of selected for, and those already been defined, or at least many of them have been. So we can set up alerts where if we're able to assemble the genome, we can just key in, hey, has this mutation been identified? And or rather, even some of these references have helped us say, hey, if you're seeing this mutation, we're already seeing adaptivity kind of play out over time. So the tools being rapidly developed has been fascinating to see.
Absolutely. And hopefully there'll be no big outbreak, but it's good to, do all this monitoring all the time for when the inevitable does come around, which is another big flu pandemic.
Yeah, that's right. I mean, I think if the history of time is any lesson, in avian flu does reassort. It may not be this case or this time, but that's why we need the resources put in play to be able to watch as early detectors of these types of events happening so that we can have public health action that curbs these types of things. So it is worth saying right now, like with H5N1, especially across North America, undeniably an agricultural impact, devastating. Yeah, I think like there was a recent report, I think in like Forbes or something, current impact across U.S. agriculture already north of $1 billion in damages in culled poultry flocks. Yeah, it's like 100+ million birds have to be sacrificed
Wow.
whenever it's detected. Yeah, so economic impact, agricultural impact already there, and as we're seeing that increased reservoir and um, spreading out into mammalian populations, that's where the human risk starts to increase. But I think currently, if you look at the CDC reports, the human health impact risk is still low, but the potential is there just because given influenza and the way it can rapidly reassort and mutate there.
Awesome. I think that's all we have for this week. But thank you very much for listening to the Bioinformatics Lab podcast, and we'll hit you up in 2 weeks' time.